For our next video lecture, we're going to continue the story of catabolism, but we're going to talk about an alternative pathway of glucose metabolism, and that is fermentation. Okay, so again, we have already talked about, let's go back to our, well, there's a little miniature version of the overall big picture diagram that we've taken a look at before, and you may need to go back and flip through your note pages to take a look at this more closely. But as we've talked about before, glucose as a nutrient sitting up here can be catabolized using different pathways, cellular respiration or fermentation. Both of those have the glycolysis pathway in common. Glycolysis converts a glucose molecule into two pyruvic acids or pyruvates. We've talked about that already. When you get to this phase, you can undergo the complete oxidation or breakdown of these pyruvic acids to carbon dioxide. That occurs during cellular respiration. The Krebs cycle or citric acid cycle runs, and so does the electron transport chain, and you make lots of ATP for every glucose molecule that you start off with. In a typical bacterial cell, you're going to get about 38 new ATPs for any glucose you start off with if you run aerobic cellular respiration. Now, what about fermentation? What is fermentation? Kind of a key thing with fermentation is it does not require oxygen gas. So if you are an aerobic organism that does aerobic cellular respiration and you don't have any O2 around, remember we talked about how that is going to wind up in a shutdown of both the Krebs cycle and the electron transport chain. If you don't have any O2 around and you do aerobic cellular respiration, all of those chemical processes basically back up and come to a halt. Glycolysis, though, can continue. Glycolysis does not require oxygen gas. So some microorganisms are able to shift over in the absence of oxygen gas and perform fermentation as an alternative. Okay, now fermentation, the ATP that gets generated is still generated during glycolysis. That's it. And if you guys remember, the output of glycolysis from one molecule of glucose is only a net gain of 2 ATPs per cell. Now, we've also talked a little bit about how there are hydrogen atoms that get plucked off of the glucose, and a hydrogen atom and an electron from a hydrogen atom bond to NAD plus molecules. Those are NAD molecules with a positive charge, and they become NADH. So they've bonded a hydrogen that was originally on the glucose plus an extra electron. And these are called electron carriers. They go off and deposit those electrons normally in the electron transport chain if you're running cellular respiration. And these are temporarily, by hanging on to the hydrogen and the extra electron, they're actually temporarily holding on to some of the energy that was originally in the glucose. All right, so we have a problem. If you're only running glycolysis, if you want to keep breaking down more and more glucose because you need more ATP over here, then you're going to need a continual supply of NAD plus molecules to keep that running. Well, there's a problem. If an organism has shifted over to only being able to do glycolysis to make its ATP, it's going to have to do a lot of glycolysis to supply enough ATP for survival, which means these NAD pluses over here are going to get used up. They're going to get depleted. So we need some way, because they're all going to get converted over to NADH. Okay, so we've got to have some way to take these NADHs that build up by running glycolysis over and over and over again and convert them back into NAD pluses. And that is where fermentation comes in. Okay, and so I know this is going to seem kind of convoluted, but what happens during fermentation is you take these pyruvic acids. Okay, now we took the hydrogens off of the glucose, the hydrogen atom and the extra electron that was plucked off of the glucose as glucose was being converted into pyruvic acids. Well, during fermentation, we're going to take those hydrogen atoms and that extra electron and stick them back on the pyruvic acids. So we're kind of going backwards here with fermentation. So when that happens, again, you got a hydrogen atom here and an extra electron are attached back onto pyruvic acids. So these guys go back to being NAD pluses, and now they can go back up here and participate in glycolysis. So it's kind of a cycle type thing that's going on between glycolysis and fermentation. All right, so when pyruvic acid receives the hydrogen atom and extra electron from NADHs, that can happen in different ways. So there are different chemical reactions or chemical reaction pathways that lead to that process occurring. And we're not going to go through the details of those different pathways. That gets into more detailed chemistry than we need for this particular class. However, when this happens, the pyruvic acids, or a breakdown product of the pyruvic acids, that's what they mean by derivative here, get converted into some type of fermentation end product or products. And these are more or less essentially waste products for the cell. And again, the whole purpose of making these, it's not because they're useful products for the bacterial cell necessarily. It's in order to provide a dumping ground for these hydrogens and the extra electrons so we can regenerate those NAD pluses. So kind of a key thing with fermentation is different organisms will accomplish this in different ways, and they're going to make different fermentation end products. And remember, all these chemical reactions that take place in cells use different enzymes to occur. So for particular bacterial species or particular yeast species, whatever it is that we're thinking about that's performing fermentation, they're going to have different enzymes or sets of enzymes that can do these fermentation reactions, and those wind up manufacturing different kinds of products. And there are lots of them out there. Fermentation end products are pretty diverse across the microbial world. And that's what we're taking a look at here on this diagram. This is listing some different genuses of microorganisms. And down here are some of the different fermentation end products they can make because of this process. So bacteria in the Streptococcus, Lactobacillus, and Bacillus genera, for example, can make lactic acid as a product. That's what our muscle cells and some other cells in our body make if they temporarily shift over to fermentation to make ATP during periods of low oxygen availability. Saccharomyces yeast will convert these pyruvic acids into ethanol and carbon dioxide. Ethanol is your drinking alcohol. Saccharomyces is a genus of yeast used in beer and wine production. So those yeast will convert sugars in grain fluids or grape juice and convert them into ethanol and carbon dioxide. And so forth and so on. There are lots of different examples that you will see here. And again, kind of a key thing with fermentation, low ATP production, so an organism has to keep doing glycolysis over and over and over again. And we can't, a complex organism like us can't survive on fermentation, but little, small, tiny prokaryotic microorganisms or eukaryotic microorganisms in some cases as well that are single-celled can get by on fermentation for a while. What they're going to need, though, is a lot of this. In order to do that, they're going to need plenty of glucose around so that they have enough of it to break down, to make enough ATP to fuel their survival. Here's another figure that shows examples of different kinds of fermentation products that are made in different ways. And again, they're reminding us here of why fermentation is done. It's to take these NADHs that come from glycolysis and convert them back into NAD pluses. But what they're showing you here is there are lots of things that we use on a daily basis as human beings that come from fermentation that you're probably not aware come from fermentation. For example, cheese production, Swiss cheese production, involves a bacterial organism in the genus Propionibacterium, and it is able to take glucose sugars, create pyruvic acid, and then the pyruvic acid gets converted into carbon dioxide and propionic acid. Propionic acid curdles the proteins in milk and converts those over to curds or solids, and that's what eventually becomes the cheese with drying and aging. And the carbon dioxide gas is what creates the holes in the cheese. Those are carbon dioxide gas bubbles. Lactic acid produced by some types of bacteria is used in cheddar cheese production, yogurt production, and soy sauce production. Soy sauce is fermented soybean fluid. Wine and beer, we talked about that. Acetone, ladies, where do you have acetone? You find that in your nail polish remover. That is actually a fermentation product from bacteria in the good old Clostridium genus that we've talked about before. So the Clostridium bacteria make that compound, and it winds up being chemically purified and sold as a nail polish remover. And there are other uses for acetone as well. Isopropanol, that's your rubbing alcohol. That's also a fermentation product. Acetic acid. Anybody know where you find acetic acid in your kitchen? You probably have some. That's diluted acetic acid is vinegar. So things like apple juice and red wine and so forth are fermented by bacteria, and they take these pyruvic acids and convert them over into acetic acid, and that leads to the production of different kinds of vinegar useful in our kitchen. So lots of products we use on a daily basis come from fermentation. Again, some of these products are useful to the microorganisms, but some of them are more or less just waste products that are generated because of the need to bring back these NAD pluses so that organism can keep running glycolysis. Okay, so in the laboratory, the fermentation capabilities of bacteria primarily are helpful, or traditionally have been helpful, with identification. So as you can see over here, there's a lot of diversity in terms of what different microbial species can make from fermentation. What products do they make? We can take advantage of that in the laboratory to help us narrow down the identities of organisms. Can this organism even do fermentation in the first place? Not all organisms can. Some can do fermentation, some cannot. If this organism can do fermentation, does it have the enzymes to start with sucrose? Does it have the enzymes to start with lactose, a type of disaccharide? When it does fermentation, can it make this kind of alcohol? Can it make this type of acidic product? And so forth and so on. So we can take advantage of the ability of some bacteria to perform fermentation to help us narrow down identities. And again, we trace it all back to what enzymes these organisms have. Different microorganisms will produce different sets of enzymes that allow them to carry out these processes. And if they can do fermentation, the enzymes they have will dictate what products they're able to produce. This is an example of a pretty commonly done fermentation test in a laboratory. Bacteria are put into a liquid broth culture medium that has a dye in it that is sensitive to pH changes. And so you're testing to see whether the organism can take carbohydrates in this medium, usually some type of sugar, and do they have the enzymes to convert that into glucose and then take the glucose and do glycolysis and do fermentation at the end. And if they can do all of that, are they making acids? So a lot of times bacteria produce acids from the fermentation of glucose, and acids have what kind of pH? Hopefully you're thinking a lower pH. The dye in this broth is sensitive to pH changes. So if the pH drops in the culture medium, this red dye that's in here becomes yellow. And so that yellow coloration lets you know, yes, this organism was doing fermentation and it was producing acids from the fermentation that caused the red dye in that culture medium to turn yellow. So that's a very traditional way of learning something about the capabilities of a microorganism and narrowing down its identity. Traditionally, those tests were done in glass culture tubes. Now a lot of these tests are miniaturized, and they're done in little teeny tiny wells and plastic dishes, and a lot of times the results are read by computers rather than from human eyes. But that's something has for a long time been taken advantage of in the microbiology laboratory. I also want you guys to be thinking about, kind of going back over here, let me go back to one of our big picture diagrams like this one. I want you guys to be thinking about, so we learned about organisms that are obligate aerobes. You must have oxygen gas for survival. So if you're an obligate aerobe, now we've already talked about you can do aerobic cellular respiration. If you're an obligate aerobe, can you do fermentation? Let's think about that. Think about that. What if you're an obligate anaerobe? Can you do aerobic cellular respiration? Is there an alternative type of respiration we've talked about that you could do if you're an obligate anaerobe? If you're an obligate anaerobe, could you do fermentation? Is oxygen gas involved in any way in fermentation? And keep in mind the diversity of microorganisms that are out there. All right, this is a diagram, and there's a similar one from your textbook. I just happen to like this one a little bit better because it's not quite as complicated. But the stories that we've been talking about with how organisms break down their nutrients and harness energy, make ATP, that story always in a biology class traditionally starts with glucose. That's our preferred sugar nutrient to extract energy from. But as you guys know, other types of organic molecules can function as sources of energy for organisms. And the way that works is that these other types of organic chemical compounds get broken down into smaller molecules that feed into these pathways that we've been talking about. So the easiest example is just to start with carbohydrates in general. Carbohydrates are multiple sugar molecules that are bonded together in some sort of pattern. And those can be chopped apart into various monosaccharides or sugars. And then cells will typically have enzymes that convert other kinds of sugars into glucose. So you undergo changes in the patterns of those molecules so that they become glucose. And once you have glucose, you can start running your glycolysis and then your cellular respiration or your fermentation as an alternative if you have that capability. So carbohydrates are pretty straightforward. What about proteins? We know we can get energy from proteins in our foods, and so can microorganisms. So what proteins are chains of amino acids. So you chop a protein apart, it's going to be broken apart into amino acids. Some amino acids can get converted into pyruvic acid, and then those can feed into the Krebs cycle and the electron transport chain. Some amino acids get converted into this compound acetyl-CoA that feeds into the Krebs cycle, and some amino acids get converted into compounds that are parts of the Krebs cycle itself. So they just wind up feeding into later parts of cellular respiration. Lipids, many lipids are triglycerides that are built from glycerol and fatty acids. Glycerol gets converted into this compound, glyceraldehyde 3-phosphate, that is part of the glycolysis pathway. Fatty acids get oxidized and converted into acetyl-CoA, which then feeds into the Krebs cycle. So kind of the big picture there is these other organic nutrients can be broken down and converted into other molecules that feed into these glycolysis and cellular respiration pathways that we have been discussing. So we just don't want to leave this story and think that living things only use glucose as a source of energy for making their ATP fuel molecules that they need for survival. All right, the last video lecture for Chapter 7, we're going to talk a little bit about the anabolism side. We've spent a lot of time on the catabolism side because making ATP is so critical, and we'll finish up the chapter talking a little bit about anabolism.